Welcome, everyone. Thank you so much for joining us today. My name is Rachel Massey, and I'm the Senior Science and Policy Advisor with CHE, the Collaborative for Health and Environment. I'm just going to go over a few quick bits of housekeeping before we get started. First of all, please feel free to introduce yourself in the chat if you'd like to. CHE is a member group of the EDC Strategies Partnership, which is bringing you today's webinar. The partnership members include Healthy Environment and Endocrine Disruptor Strategies, or HEEDS, a program of environmental health sciences, Commonweal's Biomonitoring Resource Center, and the Health and Environment Alliance, or HEAL. If you'd like to download today's slides, you can find them on the webinar page on our website at healthandenvironment.org, and we'll put that direct link into the chat now. We'll disable the chat during the presentation, but please type your questions in the Q&A box at any point, and we'll get to as many comments and questions as we can after the presentation. If you'd like to activate closed captions, you can do that by clicking the button on the bottom of your screen. This webinar is being recorded, and it's scheduled to last for an hour. Dr. Jerry Heindel will speak for about 40 minutes, followed by Q&A. I also want to mention that Jerry's co-authors are on this call as well. Sarah Howard, Dr. Barbara Corky, and Dr. Robert Lustig. Barbara and Rob will be joining in for the Q&A section of the conversation. With that, I'd like to hand it over to today's moderator, Jerrold Patton, Director of the Biomonitoring Resource Center. Jerrold?
Hi, everyone, and welcome to this really exciting webinar. I'm so pleased to have the honor to be the moderator here to talk about the Obesogen model as a unifying theory for the global rise in obesity. Now, as we all know, obesity is rising around the world at a global level, increasing. It's very scary, I think, given all the diseases we know they're connected to obesity, which include osteoporosis, diabetes, some cancers, and of course, cardiovascular disease. Today, Dr. Heindel is going to discuss 4 general models that seek to explain some causes of obesity. This include energy balance, carbohydrate-insulin balance, oxidation-reduction model, and the Obesogen model. This latter one proposes that environmental chemicals interfere with hormonal signaling. And I also want to mention today that Dr. Heindel has published an op-ed in Environmental Health News that calls out obesogens as a major cause of obesity and the need to halt exposures to these chemicals. Today, after his presentation will include a combined model that links 4 general models to create a unifying paradigm, which put in place as primary guidance for regulations and obesity prevention programs may decrease the incidence of obesity, which is badly needed. Let me give you a little bit of biography of Dr. Heindel. He has a PhD in biochemistry from the University of Michigan and worked on reproductive biology and toxicology at the University of Texas Medical School at Houston and the University of Mississippi. He moved to the National Institute of Environmental Health Sciences, NIHS, and headed their reproductive and developmental toxicology group. He then moved to the Division of Extramural Research and Training at NIHS. He was responsible for developing and administering the NIH grants program in endocrine disruption, the developmental basis of diseases and obesity and diabetes for over 25 years, and many of us are very grateful for his work there. He's now retired from NIHS and is currently the director of the Environmental Health Sciences Program at the Healthy Environment and Endocrine Disruptor Strategies at www.heeds.org. You should take a look at this very important website. Dr. Heindel or Jerry, I'm going to turn this over now to you for your presentation and for your slides to start working with us. Thank you very much.
Thank you, Jerrold. I appreciate the nice introduction, and I'm really pleased to be here today with my co-authors to present to you our new model of obesity. And the reason we're here today is that this slide should change there. Whoop. I know. The reason we're here today is because of this data, what we have here, the trends in obesity, not just in the United States as shown here, but across the globe. The increase in obesity in both men and women over the last 20 or 30 years. And what's even more disturbing is the increase in children and adolescents, including the 2 to 5-year-olds. So this is a major global problem. And I'm going to approach it by asking and answering a few questions, the kind of questions hopefully you've been thinking about yourself. The first one is, what causes obesity? Well, guess what? You may not understand exactly what causes obesity, and that's okay because the scientists don't either. Here's some statements that have come out recently. The current lack of consensus regarding obesity pathogenesis has resulted in competing and poorly justified claims, both from within and outside the scientific community. And this one, we don't have a clear explanation for the obesity epidemic. And finally, they're still working on defining what obesity is. So there's a lot of work still to do here. But the one thing we do know is that obesity is quite complex. So it's due to a lot of things. You can intake too much food, changes in energy expenditure, exercise, environmental impacts, lifestyle factors, and genetics. Although we do know that this increase over the last 30 or 40 years can't be due to genetics per se. So it's due to these other factors, and we're going to look at those today. So let's look at when obesity actually starts. Well, what's interesting is that 6-month-old babies are becoming obese, and even newborns are affected, the increased weight. So that says that really any hypothesis we're going to propose here has to be able to explain why newborns and young babies are becoming obese, because it's not explainable by diet and exercise alone. So what we know is that these critical periods have to be accounted for. So while nutrition, stress, and environmental chemicals can cause weight gain across your lifespan, we know now that development in utero in the first few years of life is the most sensitive to their effects. And this actually all started many years ago by David Barker. It was called the Barker hypothesis, and then the development origins of adult disease. And it's focused on nutrition. During the Dutch hunger winter in World War II, the Nazis cut off food, and the women that were pregnant there were malnourished, and they had babies that had low birth weight. And when they followed these babies to adulthood, they found they had increased heart disease, hypertension, diabetes, and obesity. So this whole developmental origins started with nutrition. But we now know that there are other methods, not just nutrition, but stress. And as I'm going to show you, environmental chemicals can play a role here. And here's how this goes. All cells contain the same DNA, but during development, there are hormones and growth factors, and they turn genes on and off so that you can form specific cells like a liver cell or a kidney cell. And that's called epigenetic programming. And these environmental agents can alter this epigenetic programming. And the result then is that these cells and tissues have altered genes turned on, or protein function, or cell numbers, or cell location. We call that functional changes. And that leads to a tissue that really basically looks okay, but when you get down to the molecular level, as I showed here, there are many changes that permanently alter how it functions. And these changes then persist throughout life. And the effects of these changes can occur across the lifespan and may actually require a second hit. So these programming changes then lead to increased susceptibility to a variety of diseases. So we call that a bad start lasts a lifetime. So what diseases are affected by developmental exposure to environmental chemicals per se? You can see all these diseases are affected, but today we're focusing on the fact that metabolic syndrome and obesity, but also nutrition, overnutrition or undernutrition can also lead to obesity, diabetes, and fatty liver disease. So this has to be taken into account in any model of obesity, that developmental exposure to chemicals or nutrition can result in these diseases later in life. Now, one more thing before we jump in, it's important to understand that this whole metabolism is controlled by hormones. This whole complicated, integrated system here is all hormones talking back and forth between each other and between the brain. So, and the brain is the controlling mechanism here. It controls your appetite and satiety by specific neurons. And it also contains, it, it, it plays a role then in reward mechanisms or craving, your emotional eating and addiction. So both parts, whether you should eat or not, or you really want to eat or crave eating, are controlled in the brain, and they're controlled by all of these different tissues and the hormones that these tissues put out that act on each other. And you can see all the arrows here going to the brain. So wherever there are hormones controlling here, that means those are potential targets that can lead to obesity. Okay, so let's go into the current models of obesity. So there are two models that are pretty well accepted by the scientific community as playing some role. The energy balance model that basically says obesity is due to overeating. It doesn't matter what you overeat. If you have more calories, you will gain weight. And it also says then that it's the brain that controls weight gain that's responsible here. The other model, called carbohydrate-insulin model, says it's not just any calories, but it's sugar. Sugar then stimulates insulin, and insulin stimulates fat storage. So let's look at each of those in the little schematic. These are very simplified, but give you the basics. So the energy balance model says you overeat, you have increased energy intake, that increases your metabolic fuels, and you put in more fat and you get weight. And this overeating, or the control of eating, is unconscious through these appetite and satiety neurons and these emotional addictive controlling neurons. And the bottom line is it's this ultra processed food, energy dense, ultra processed food that affects this hedonic and homeostatic control mechanism and causes you to unconsciously really overeat, leading to obesity. So in a simple form, that's that model. And here's some clinical data that says ultra processed foods cause weight gain. So they gave people ultra processed food or unprocessed food that have matched for calories and sugar and fiber and everything. And over a 2-week scan, you can see right away the ones that were eating the ultra-processed food ate more food and they gained more weight. So this ultra-processed food can affect your brain controlling mechanisms and stimulate you to eat more. So you're probably wondering, what are ultra-processed foods? Well, there's a system that's been developed called NOVA food groups. And if you look down here, this is all the great stuff you like cheeseburgers and breakfast sausages and pancakes and biscuits and candy and chocolate, hot dogs, ultra processed things that are made from a variety of things. So there's support for this model because you can lose weight. It doesn't depend on what diet you're on. So it's just calories that count. And because of what I just showed you, this ultra processed food can cause weight gain. But there are still some challenges. Why have animals also gained weight? What about the increased weight of babies? And what about this developmental program that I talked about? They don't talk about that at all. And they don't really have a specific mechanism to explain the altered control of food intake. So there's some more work to do here. So let's go to the second model, and it's going to focus on insulin. And just as background, we all know insulin comes from the pancreas. It's an anabolic hormone, so it stimulates glucose uptake into cells. It stimulates fat synthesis and deposition, and inhibits fat release and oxidation. And if you have excess insulin, it will cause weight gain. And there are things you can do. If you decrease insulin action, you can cause weight loss. So certainly insulin plays a role here. So this is the carbohydrate-insulin model. It says it's not food in general that's the problem. It's the high sugar content of food. The sugar that stimulates insulin. And if you stimulate insulin, you create fat. And at the same time, then you have the glucose and lipids going into fat. They're decreased in the blood. That goes to the brain, tells the brain that you need to eat. And so you get increased fat, increased eating, and obesity. So there are data supporting this model. I think we all know that ketogenic and low carbohydrate diets seem to be the best ones. They're better than low fat diets. And if you inhibit insulin secretion, you can cause more weight loss. And this Western diet certainly stimulates insulin. So it's likely this could play a role in a subset of individuals. But there's no focus on developmental programming here. And these high glycemic processed foods probably contain many other things that may stimulate insulin. So there's still a little more work to do here. But really, if you take a look at it, both of these models make sense. If you increase your food intake, you have positive energy balance, increases fat mass. If you have high glycemic load, you increase insulin, increases fat mass. So both of these models work to some extent, at least. And so it's kind of silly to say it has to be one or the other because they both have a role, I believe. Now, what about environmental chemicals? That is another model that we want to get into, and one that's not as well known. So these are obesogens. we're going to talk about, and obesogens are chemicals that alter energy metabolism. They alter the signaling pathways in various cell types that regulate metabolism and energy expenditure. So they can be all kinds of obesogens. Metals and viruses work. Some prescription drugs put on weight. A lot of different environmental chemicals, which is where we're going to focus, and even some food components. And the majority of these, especially the anthropogenic, environmental, and food components, are endocrine disruptors, and that's how they work as obesogens. So let's put that in context for you. There are a lot of chemicals in commerce, and some of them have been tested and found to be toxic, and some are toxic because they're mutagens. They alter DNA, or they just generally kill cells. But some are toxic because they interfere with the endocrine system, and they're called endocrine disruptors. So an endocrine disruptor is an exogenous, either chemical or a mixture of chemicals, that interferes with any aspect of hormone action. Basically, what that says is these are chemicals designed for something, like a pesticide, perfectly normal and legal, but they have a side effect that they mimic or antagonizing the hormones that are controlling metabolism or some other aspect of the body. And if they mimic or antagonize hormones that control metabolism, then they're called obesogens. So obesogens are a subset of endocrine disruptors. And here are some obesogens that we're exposed to. I think we're all familiar with the bisphenols like BPA in water bottles, canned food, phthalates that are in cosmetics, food packaging, medical devices. They make plastic more malleable. Perfluorinated chemicals, nonstick cookware, water-resistant clothing, stain-resistant fabrics, PCBs, which are lubricants and transformers, organotins in paints, dioxins as byproducts of industry, PBDEs, which are flame retardants, and parabens are preservatives in personal care products and some foods. So these are all endocrine disruptors with obesogenic properties, and we're all born pre-polluted with a variety of these. And indeed, bisphenols, phthalates, and PFAS are in basically 100% of us. Now let's go back to our endocrine hormonal control of mechanism slide. And basically what we're going to see is that all of these different tissues that are producing hormones and affecting the brain are targets for obesogens, because obesogens are chemicals that target hormones that control metabolism. So they can control your appetite and satiety, your food addiction and craving, and alter all of these different tissues. This is our kind of more complete list of obesogens: smoking, nicotine, air pollution, DDT, and then here's our perfluorinated bisphenols and phthalates, which come from fossil fuels, tributyltin, flame retardants, non-nutritive sweeteners, agricultural pesticides. But here's one you probably never thought about: food preservatives, additives, and emulsifiers, the kind of things that you find on the package labels, monosodium glutamate, parabens, and some of these others. These have been shown to be obesogens. So here's just a couple of quick examples. So what you do in this experiment is you expose pregnant mice to BPA in utero and the first few weeks of life, and then you look at the offspring, and here's the control offspring gaining weight, and the ones that had BPA increase weight gain. Now here's another model of BPA. This is a rat model, and similarly, it increases weight. But in this model, they have actually shown that it increases food intake as the mechanism. So they went and looked in the brain, and guess what? They could see there was more appetite stimulating neurons and less satiety neurons in the brain, an actual mechanism for the increased food intake due to developmental exposure to BPA. BPA also makes can make fat cells contain more fat in vitro. These are pre-fat cells, the fat cells. Phthalates are obesogens. This is one specific phthalate. As an example, these are adult mice treated for 8 weeks. Here's the control, and look at all this fat due to phthalate exposure. And it's visceral fat, which is bad fat in humans, and it also increases food intake. And it also works if you do developmental exposure. Same thing, do developmental exposure, look at 8 weeks, and you see the increase in fat. Higher body weight, increase in visceral fat. So I don't have time to show more data, but there are extensive studies, like 18 studies showing phthalates in human population, a large study showing BPA exposure causes obesity in children and adults, and BPS, and large pooled analysis of air pollutants, and a systematic review and meta-analysis of 33 studies that said DDT and hexachlorobenzene were both associated with childhood obesity 2 to 9 years. So there's quite a bit of human data supporting the obesogen hypothesis, developmental exposure, and weight gain later in life. Now I want to go back and look at this Western diet. This is the diet that many of us are on: high fat, high sugar, high salt, low fiber, inadequate fresh fruits and vegetables, micronutrient deficient, high in ultra processed food, and is addictive. So certainly this is the ultra processed food diet that we're on that was just shown to cause weight gain. But did it cause weight gain just because of this? Well, guess what? This Western diet also contains specific obesogens. Here's the bisphenol A, phthalates, and perfluorinated chemicals again, and they leach from the plastic that these are stored in. Then you have fructose, and you have all of these non-nutritive sweeteners, parabens, and monosodium glutamate, and additives, preservatives, and even some food colorings. And then you have pesticides that were sprayed on the fruits and vegetables that are still there. So this is a really quadruple, double whammy for putting on weight. The diet itself and all the obesogens that you get in it. What could be worse than that? And here's a huge study. Look at all the authors. Shows it causes weight gain in adults. It's not only weight gain. This diet is so terrible. It causes Crohn's disease, asthma, alters your bone, cancer, dementia, mental health. It is so terrible. but it's the diet of the day. So I don't have time to go into specific data, but obesogens can modify all of these different endpoints. Pretty much all of the endpoints that I showed on the slide, where all the different tissues were integrating with the brain, they increase the number of fat cells, they cause fatty liver, alter the microbiome, cause oxidative stress, increase food intake, food addiction, decrease energy expenditure. Everywhere you look, these obesogens can have an effect. And here's another way to look at it. Look at all the different obesogens that have been tested and found to alter the microbiome. And these affect increased food intake. And all of these have a larger effect if you have a high-fat diet compared to a normal effect. These non-alcoholic fatty liver disease, some of them make not just more adipocytes, but they make dysfunctional adipocytes. And this is important because here's a long list that increase reactive oxygen species. So we're exposed to a mixture of all of these chemicals. And for example, if you're exposed to BPA, which we all are, look at all the different things that BPA can do to stimulate you to put on weight gain. And similarly for some of these others. Now, I want to make one last point about obesogens. And that is that obesogens can act across generations. So this is the typical experiment that we've been doing and that I just showed you. You expose a pregnant animal to some chemical, and then you analyze the offspring for weight gain. But if you take these animals and mate them down, and then mate these to the next generation and these to the next generation, you get a generation here that has never seen the toxicant that was put in here that caused weight gain here. And if there are effects here, they had to be transmitted through the sperm. And look at here, positive results, transgenerational, over 4 generations, tributyltin, DDT, BPA phthalates, and even nutrition. So here's one last little piece of data that I just think is so fascinating. It makes a very important point. And this is tributyltin, and it's the 4th generation that has never seen any tributyltin. And if you look right here, this is the control and the ones that had tributyltin, their body weight, nothing going on. But if you give these animals high-fat diet, you can see that the ones that had tributyltin 4 generations before are sensitized and they put on more weight. And then you take away the high-fat diet and they have a harder time losing their weight. Now, does that sound familiar? You gain weight more easily and harder to lose weight. And one more, if you put these animals on fasting, the controls lose weight and the tributyltin don't wait. So they're resistant to fat loss. I just think that's so amazing. 4 generations later, and it shows the human situation today. So here's the obesogens. They interfere with these hormone signaling pathways in all these tissues, causing all this variety of effects that I just showed you, leading to fat storage and weight gain. Now, here's only one clinical kind of study that we have, and I think it's very informative for several reasons. One is they gave 4 different diets for 24 months. And guess what? People lost weight on all 4 diets. It didn't matter which diet, they all lost weight. But then they continued through 24 months and they gained weight back. And that was the same with the diet. But the ones that had perfluorinated chemicals at high concentrations before the start of the study, they gained the weight back greater. And it was because they had a lower metabolic rate. So their metabolism was slower, so they gained more weight. And that's one way, again, that obesogens can cause weight. So there's a lot of data supporting the obesogen model. Development and adult exposures work. There's exposure, and the endpoints, as I just showed you, are similar to the ones that they say are important in the 2 other models. Still, there's some work to do because we don't really have a biomarker that we can look at in adults and say that there was exposure in utero. That would be really nice to have. And we need more clinical kind of studies like the one I just showed you. Now, there's one last model I'll go through quickly, and it's called the REDOX model, and it's something you probably never heard of. But this is an energy responsive communication system that changes with the fuel needs of cells. So the normal response to fuel in a cell is to transiently increase these reactive oxygen species. And that is in the cell, and they go in the blood, and they signal that there's enough fuel. They increase insulin, alter brain control of food intake, stimulate triglyceride synthesis in adipose tissue. But if the signal is inappropriate, something comes in and stimulates ROS when it shouldn't be, then you have an inappropriate signal, and that causes problems. And I'll show you that. So here's the normal situation. Normally, you just have regular food consumption. The cells are very happy. They have plenty of fuel, as shown by increased NADH in the mitochondria. That gives you a little burst of ROS, which increases insulin to make storage in fat and liver, decreases food intake, and decreases gluconeogenesis, and all is good. And these symbols then, ROS and some other metabolic regulators, go in the blood and they talk to each other, so they're in a coordinated manner. So what has changed in the last 50 years that could result in inappropriate ROS that could cause a problem? Well, as I showed you, and I'll show you again, obesogens are, many of the obesogens will stimulate ROS, and ultra-processed foods stimulate ROS. So let's look at the metabolic disorder. So ultra-processed foods, excess food consumption, and obesogens both then give an inappropriate signal and give you a huge increase in ROS that's not normal. That stimulates hyperinsulinemia and actually increases food intake and causes hyperlipidemia. So you get increased fat and adipose tissue inflammation. So this is a nice signaling pathway that should work normally, but with ultra-processed food and obesogens doesn't. So both the OBS and redox model actually go together very nicely, because the ultra-processed food and obesogens change the redox. and that creates a misleading, inappropriate signal and causes these problems. Now, the redox model is largely hypothetical, so while there's some support, there's still a lot to do. You need more evidence that you can change ROS in human circulation and their ability to generate ROS by specific chemicals, for one thing. So the first thing we did then, take a look, we said redox model and the OBS model actually go together because both diet and obesogens cause an abnormal increase then in ROS. And that abnormal increase in ROS goes to these tissues, causes increased hunger, increased insulin, and increased fat. So those two go together nicely. Now, what about the other two? Well, we have diet and obesogens. We believe that obesity is due both to diet and obesogens. The ultra-processed food diet, overeating diet, and they work because they stimulate ROS inappropriately as a system that controls metabolism. They interfere with the normal hormones system that controls metabolism and lead to weight gain. But what about what's happening here? Well, this is where it all comes together because obesogens stimulate both of these mechanisms, and they both then stimulate all of these endpoints that I showed you, the endpoints that are noted in the energy balance model, increased food intake, altered brain control, and the carbohydrate-insulin model. So you didn't have to put these two models there. Whoops, let me go back. You don't have to put these two models there. You can just put these and you have the weight gain. So the obesity model now can focus on obesogens out here and weight gain over here and all the endpoints that take both of these models into account. So the key points, the cause of obesity is unknown and it's very complicated. There are these 4 different models. Each model, we believe, makes essential contributions to the pathogenesis of obesity, and we shouldn't be arguing one over the other, but try to look at how they work together. And we propose this integrated model that can explain all of the major changes that are responsible for weight gain in the other models. And here's all of these that I've gone through already several times. And our model puts the focus then on obesogens because obesogens can do all of these things. So that's the first thing that's key and different. It puts a focus on obesogens as playing a major role. The second key thing then is if you accept this model, it changes the whole way you look at obesity because now we can focus on preventing obesity by reducing exposure to obesogens. And that's really critical. That changes the narrative to prevention. So there are some personal changes that you can do. Reduce your exposure to environmental chemicals. And you want to do that before and during pregnancy. And then, of course, throughout life. And there are things to do here, including eliminating plastic and ultra-processed foods. But you can't solve the obesity epidemic at a personal level. You can do what you can for yourself, but really it's a public health problem. And you have to be able to provide affordable, sustainable, healthy food. You have to be able to reduce exposures to obesogens. And to do that, you need policy and regulatory changes. So we hope our focus on obesogens will help move all of these things forward. So we're certainly not at the end, but we're at the end of the beginning of solving the obesity pandemic. And thank you very much.
We do have some questions here, and I believe, Rachel, we're going to ask Bob and Barbara to answer these questions. Is that correct?
Whoever wants to speak up and take the question, that's fine.
Okay. What does the science of obesogens tell us about the potential benefits or harms of drugs that are used to treat obesity, or are these just two unrelated topics? This is the first question I have.
That sounds like a good one for Rob.
Hi, everyone. So the bottom line is the drugs that are being used to treat obesity are not dealing with the problem. They are dealing downstream of the problem. Yes, they are effectuating weight loss. In particular, of course, GLP-1 analogs have sort of taken over the space. You almost never hear about any of the other anti-obesity medications. The question is, why do they work? Do they have anything to do with ROS? Do they have anything to do with endocrine disruption? No, actually, the reason they work is because they delay gastric emptying, and you eat less because you don't move the food through the intestine faster. That's one of the reasons for the nausea and the vomiting, and also for the gastroparesis and the pancreatitis. So while they do work, they are reducing food intake. So I guess in that respect, they're dealing with the energy balance model, but they're not altering mitochondrial beta oxidation. They're not altering reactive oxygen species generation. So in essence, they are a Band-Aid for a different problem. Do they work? Sure, they work. But at great cost and at great expense to the patient and to society.
And it turns out that when you stop taking them, the weight comes back again.
Exactly.
So that
right.
really is a Band-Aid.
It's a Band-Aid.
Can you talk about how obesogens impact men and women and boys and girls differently? Is there any kind of difference between genders?
So obesogens affect both men and women and girls and boys, but just hormones are different in men and women. And so some of the effects are different in men and women. Some of the obesogens have stronger effects in females than males, and some are the opposite.
Okay. Is DHP the only phthalate that's an obesogen, or are there other phthalates that are could have the same kinds of effects as DHP?
Now, there are other phthalates that are obesogens.
Jerry, you should take that.
I'm sorry, I don't know offhand which ones.
Okay. Are there known ways how EDCs alter microbiome? Thanks for bringing up the update on obesogens. And thank you, Jerry Heindel, for your excellent predation. That's part of the question. But can you talk about EDCs and the microbiome?
So I'll take that. I don't think there are any data on chemical EDCs in the microbiome. There's an enormous amount of data on ultra-processed foods in the microbiome. And since ultra-processed foods are EDCs, that I can very specifically talk about. There's no question that through the addition of fructose and the lack of fiber, and also the addition of emulsifiers, those three things alter the microbiome in very specific and stereotypical ways. They reduce microbial diversity, they increase gut inflammation, they reduce the mucin layer, which is one of the physical barrier of the intestine, thus increasing gut inflammation. Cytokines end up from the gut into the portal system, bind to NADPH oxidase in the liver, activate that, increase ROSs, and you're off to the races in terms of insulin resistance and also increased food intake. So without question, the EDC aspect of ultra-processed foods through changes in the microbiome are only contributing even more negatively to chronic inflammation and morbidity and mortality. I don't know about specific EDC effect, shall we say, chemical EDC effects like chlorpyrifos, or I assume chlorpyrifos, because it's a pesticide, has effects on the microbiome, but I don't know that that's been studied directly.
Okay.
I showed the one slide where I showed there were maybe 10 or 12 different obesogens that affect the microbiome in some way or another. We haven't looked at those in enough detail to know mechanisms, but they all have some effects.
Okay, thank you for that. Is the food industry at all open to addressing obesogens, or is it actively working against efforts to reduce exposure to obesogens?
Barbara, you want to take that, or should I?
You can have it.
Let's put it this way, there are two food industries. There's the CPG industry, the consumer packaged goods industry, and they don't want to do anything because they don't want to disrupt their gravy train. And then there is the upstart food startups. In fact, Future Food Tech is going to take place here in San Francisco in 2 days, and I'm actually one of the speakers. And there are a lot of people who are interested in disrupting the current supply chain by providing healthier options and using technology to try to reduce things like obesogens. One of the big ones, of course, is sugar reduction. And there are 5 different ways to improve sugar, the sugar footprint. There's reduce, replace, remove, boost, and block. And I'll be talking about all 5 of those on Friday. So yes, there is some movement, but not from the labels that you know.
Okay, thank you very much. Barbara, did you want to add anything to that?
No, I'm completely in agreement. And I think the biggest problem we have is the need for central regulation that will affect all manufacturers and all of the ingredients in our food. And I think, I believe that France has outlawed the use of plastic packaging, which I was very excited to hear, because it's that kind of an awareness that we need to begin. We don't have it here.
Not yet. Let's say not yet, Barbara, and be optimistic about the possibilities of the United Nations of Plastics Treaty that may get rid of some of these plastics floating around. We shall see what happens. A couple of other questions here. Transgenerational epigenetic transfer, does that mean generation 4 is more sensitive to further exposures to obesogens? Are they more sensitized because of in utero exposure in the previous 3 generations? We know that.
That's what I showed in that slide. But outside of that data from one lab, which actually now has been repeated 3 or 4 times, so we're really solid data that there's that increased sensitivity in the 4th generation. They haven't done any more experiments, which we really need to know how sensitive they might be to adding tributyltin or some other obesogen back at that time.
Okay, thank you. Nanoplastics we inhale every moment may act as vehicular obesogens, but are not regular in terms of their concentration. This is simply a statement. Is there anything that you would add to that comment?
So there's certainly a lot of interest in nano and microplastics as toxicants. And there's some data, but not very much yet, that they actually, in the animal model, can cause weight gain. But I think that that's the future. And as you just said in that statement, it's the particles themselves, plus the fact that they glop on all kinds of other toxicants that stick to them.
Okay. A couple of more questions. We're getting close to the hour, but we have a few more questions here. Someone's asking a little bit more explanation about ROS. It's a term that we're not familiar with, I think many people. And Jerry, you mentioned they were a species. So can you describe what ROS actually are and the increase of them you already talked about in terms of inducing obesity, but ROS themselves, we don't quite understand what they are.
Yeah, I didn't understand it initially either, and it's that's why we have Barbara to help us here.
Hey, Barbara.
Okay, so I'm a basic scientist and I'm very focused on understanding the mechanism by which these things might work. And because there are so many different things that cause obesity, I began looking at very basic mechanisms and what might be involved. And everybody knows what inflammation is, or at least they've heard the term. But when you say inflammation, it means ROS is being produced. That is what the term means. So I'm using a much more descriptive, that's reactive oxygen species, and it has a particular conformation. But this is a very important signal in the cell. It is what signals satiety. It is what happens when you've had enough to eat in every cell of the body. And it is what most of the obesogens that Jerry mentioned have been shown to cause inflammation, which means they generate ROS, not necessarily by the same mechanism. But the value of looking at this very common thing is it gives us a way to test. So how do you know if something is an obesogen? How can we know what to remove from the air and from our diet? And if you have to do a long-term animal study with feeding, it becomes so astronomically difficult to think about it. But if you have one molecule you can test for, and that proves to be an indicator of the disruption that we're talking about. And I believe that the most worthy candidate for that model that we know of now, based on current research, is ROS. So it's easy to take a cultured cell and do 1,000 or more tests at the same time with existing chemicals. It's not being done. So yes, work needs to be done at the government level, but work needs to be done at the basic level so we can, in the future, hopefully prevent these things from coming into our environment. So ROS is just an indicator of sufficiency. It's a signal, it's a good signal, but it's like everything. I think Jerry mentioned that in obesity, in most obese individuals, insulin levels are much too high. They're sometimes 9 times normal. And then these people develop diabetes and they say it's a disorder of glucose metabolism. Well, no, not really. Why is that insulin high for years and years and years? And eventually, diabetes is the consequence. Well, ROS stimulates insulin secretion when no glucose is present. So it's a very good candidate for that. So understanding ROS, it's not so important for most people in the public, but it is important to know that research needs to be done that will allow us to identify harmful things. And we need to have simple ways of doing this that can be done rapidly in large numbers.
Thank you. That's very helpful. I appreciate that your explanation of that. And of course, everyone is exposed to a whole jumble, a whole mixture of obesogens at any given moment, from air, water, soil, whatever. It's just, it's a soup. It's terrible. Yeah. What recommendations might you all offer to the future of U.S. dietary guidelines?
I could do
Get the sugar down and get the fiber up is a good start.
Okay.
I think I think we need to support research into this most important area, that the number of dollars per person that we spend on other diseases is many, many, many fold in excess of what we spend on the metabolic diseases like obesity and diabetes. And yet, as of current times, we have 3/4 of our population is overweight or obese, and this isn't going away. So we need to spend more money at a basic level.
That's really a stunning figure, isn't it? That obesity has risen to such levels. Are there any theories regarding the commonalities of all these various chemicals? At first look, they look mostly appear to be hydrocarbons and or lipophilic. Are there shared characteristics between all the or among all obesogens?
Well, the shared characteristics is that they mimic or antagonize hormones that control metabolism. And there are a variety of different hormones act through a variety of different receptors, and they either act like those hormones or antagonize those hormones. That's what makes them obesogens. So the structures can be very different, but because they act on different hormone receptors.
Right. One way to think about this is that there are three primary checkpoints in each cell that determine whether or not any given cell will burn or grow and whether it will store. Those three are all kinases: AMP kinase, PI3 kinase, and mTOR. Each one of those is a kinase. Well, when the reactive oxygen species burden within any given cell goes up, those three kinases change phosphorylation status, which means they change their energy pathway. And so you can go from burning to storing just by the accumulation of ROSs. Now, the cell has an ROS detoxification mechanism. It's called antioxidants, and it occurs in the peroxisome. But if you don't have enough antioxidants because, for instance, you ate ultra-processed food, then you can't detoxify those ROSs, and so the adiposity will continue. So understanding the relationship between food, ROSs, and their clearance and how that affects energy metabolism really explains all of obesity.
It's extremely interesting. We reached the end of the hour, a little bit past. Rachel, I think it's probably time for us to thank our excellent presenters and close this particular session. But I want to thank everybody, all the good questions. And thank you, Jerry, for this incredible slideshow that you've presented. And of course, Barbara and Bob, your excellent interventions here were extremely useful. And if it were up to me, I could be asking questions another hour at least. And I know other people and participants would feel exactly the same, but we did promise to end at this particular time. So thank you very, very much. And we hope to be back and talking to you some other point. So thanks very much. So Rachel, I'm going to turn it back to you.
Great. Thank you so much, Cheryl and Jerry. That was a really excellent talk. Thank you so much. And Rob and Barbara, I'm really glad that you were able to join us for the questions and answers. This is extremely informative. Thank you so much for taking the time to talk with us about it today and for all this really important research that you're doing. So I'm just going to quickly wrap up with brief announcements. A video recording will be available on Che's website soon, and you will all receive an email with a link to the video. We encourage you to support the work of any and all of the groups involved in the EDC Strategies Partnership. You'll find links to each group on Che's Partnerships page. If you appreciate these webinars, please consider a tax-deductible donation to Che and/or any of these partners. We also invite you to join us for upcoming webinars. On March 28th, the next Che Alaska webinar will be on chemical exposures and children's mental health, and I pasted the link for that webinar into the chat just a couple of minutes ago. So with that, we'd like to thank you again for joining us today. Thanks again to Cheryl, Jerry, Rob, and Barbara, and to Sarah Howard as well. And many thanks to Che Director Kristen Schaefer for all of her help behind the scenes.